This is a lecture on plant senescence from October 12, 2020. And in this lecture, we're going to discuss some of the terms associated with plant senescence, such as the difference between programmed and stochastic senescence. We're going to talk about the roles of these different hormones in plant development as well as senescence, and then we're going to look specifically at leaf senescence and talk about the changes that occur at the macro levels and the micro levels that lead to these three stages of leaf senescence, as well as the role that sugars play in this whole process. And so understanding how plants senesce is important. And the main focus of this class has been sort of animals and humans and aging in those organisms. But a lot of what we know about kind of the cellular mechanisms of aging have at least begun to be studied in plants, specifically leaves, right? And so leaf senescence is important in plants because it needs to occur in order for seeds as well as for fruits and vegetables to ripen. Because during senescence, the nutrients that are lost from the leaves are transferred to parts of the plants that we actually eat, right? And there is evidence that genetically engineering plants to have either a delay in senescence or to have extended senescence will yield higher amounts of crops, as well as a higher nutrient content in the seeds and fruits that we actually eat, right? And that's because during senescence, leaves sort of become a sink for nutrients, right? And this also has sort of ecological value. And during abscission, or the regulated separation of leaves, those nutrients that are now trapped within leaves will fall to the ground, right, after they're abscised from the tree. And those nutrients can go back into the soil for the next generation of plants. And so understanding what's happening at the molecular kind of cellular levels during leaf senescence allows us to understand how plants ripen so that we can modify them, as well as how nutrients can actually be transferred ecologically to become a source of nutrients for other plants. And ultimately, plant development and growth and senescence are all regulated by a series of hormones. And the most important ones in terms of this class, in terms of senescence as well, are listed here. Starting with abscisic acid, auxins, cytokinins, ethylene, and gibberellins. And you can see the types of hormones that these are here, right? And some of their main functions. And you'll notice that there's a function for senescence in almost each one of these, right? And so we're going to talk more about the specifics of these hormones a little bit in this lecture, and then as well in the subsequent one. Right, and so one of the main groups of plant hormones are auxins. And auxins are kind of a group grouped into the growth hormone category, because what they do is they promote growth of apical buds, or the buds at the top of the plant, and then they inhibit growth laterally, or these lateral buds on the sides. And the way that they actually do that is by increasing in their concentration at the apical part of the plant and decreasing in concentration near the lateral buds. Auxins, importantly, in terms of senescence, can actually inhibit the decay of chloroplasts, which will become important as we talk about sort of what's happening during leaf senescence. But this inhibition of chloroplast decay actually inhibits leaf senescence. And so that sort of makes sense, right? So as a growth factor, auxins actually seem to inhibit senescence because they favor growth. The gibberellins are kind of a large group of hormones. There's about 100 of them that have been discovered at this point. And a lot of their functions have to do with promoting kind of either elongation of plants or stimulating germination of seeds, inducing flowering in plants, And so we'll talk a bit more about how these hormones are involved in senescence later in this lecture. But what's important to note is when we think about senescence in plants versus in animals, they seem to have evolved kind of differently, right? And so we talked about how in animals, senescence and longevity likely evolved through sort of a trade-off mechanism, right? Or antagonistic pleiotropy, where genes involved in extending longevity and improving aging were selected for prior to reproduction, and then have effects that are negative that appear after reproduction, but therefore can't be selected against, right? And so you sort of trade off, right, and select genes that are going to be good to help you get to reproduction and ultimately pass your genes on to the next generation, but that they seem to have negative effects later on that you can't really get rid of, right? And a lot of aging in animals as well is kind of driven by this random process, right, or an increase in entropy that's just occurring in the universe with time. But in plants, senescence isn't a random process. It's not a consequence of selecting for genes that increase fitness. Senescence is actually an important part of the reproductive life cycle of the plant, right? And it keeps the plant alive by basically changing where the nutrients are organized, right? And so senescence in plants is highly regulated and it's highly controlled. And about 25% of all the genes within a plant are dedicated to this process of senescence and making sure that it goes correctly so that the plant can live for another reproductive life cycle. There is some senescence that happens in terms of kind of this cellular level, right? Our proliferative senescence, or the stopping of cell division. And so plant meristems are groups of undifferentiated cells that are usually at the tips of roots, as well as at the tips of shoots or the top of the stems, as well as these kind of lateral meristems that allow plants to grow wider, right? And what's interesting about plants, unlike animals, is that their body plan is determined post-embryonically by their interactions with the environment. An animal's body plan is its body plan, right? It's going to happen and grow in the same general way every single time, as most of its body plan is determined by its genetics. Whereas plants can sort of change their body plan by interacting with the environment, right? And they use meristems, or these groups of mitotic cells, to do that and grow in particular directions, either outward, laterally, or up and down at the apical meristems. But ultimately, these undifferentiated cells will go through a proliferative senescence, meaning that they will stop dividing in the same way that we see with animal cells. And in animal cells, we know that the kind of decrease in replicative or proliferative senescence is usually linked to telomere shortening, right? And the mitotic clock theory kind of suggests that the shortening of telomeres leads to proliferative or replicative senescence in stem cells or undifferentiated cells. However, in plants, that mechanism that triggers proliferative senescence is relatively unknown. But what we do know is that it's not telomere shortening. Telomeres actually sometimes even get longer, the longer that a plant lives, but they don't actually get shorter. They'll either stay the same, potentially get longer. And so that has nothing to do with the proliferative senescence seen in plant meristems in the same way that it does in animal cells. And so wild plants do have these meristems, or these populations of undifferentiated stem cells. Those populations are relatively small. And like animals, most of the cells in a plant are postmitotic, right? Or they are no longer undergoing cell division. And so they're also subject to senescence. Not replicative or proliferative senescence, where you stop dividing, but the age-induced sort of dismantling of cells that are post-mitosis or not dividing anymore. And there are two phases of senescence in plants. There's a programmed senescence, which is age-induced, and it's regulated. And it basically is just a degradation of postmitotic cells that begins with the chloroplasts and other organelles. And then there's a stochastic senescence, or the random degradation of plant cells. And programmed senescence comes first, and then is regulated, degrades these particular organelles, and it's followed by this random degradation, or stochastic senescence. And that is where the nucleus of the plant cells break down, the central vacuole, which is the major storage unit of the plant cells, breaks down. It releases some toxic compounds to the cell, and the plasma membrane can actually develop holes. And the more that this process takes place, the more holes the plasma membrane develops, and it ultimately loses its ability to be selectively permeable. And so the cellular processes get disrupted, chemical gradients are disrupted, and cell death follows soon after stochastic senescence. And when we study senescence in plants, really what we study the most and what has kind of the most mechanisms associated with it that are well understood is leaf senescence in particular. As leaves are a large part of the plant, we can watch them change at the macro level, so you can see a young and an old leaf here. And then we can also look at the micro level, because leaves generate a lot of tissue that you can study, as well as a lot of cells, right? And the main plant that at least began studies in senescence is the kind of genetic model organism for plants, which is Arabidopsis thaliana. And you can see this picture, the cute little Arabidopsis flowers here, and then the whole plant down here, and these are actually Arabidopsis leaves. And so much of what we know about leaf senescence comes from studying Arabidopsis. And we're going to talk about the phases of leaf senescence and how sugars are involved in that process in the next lecture.